Alright, so we're back here again with Chapter 3 on microscopes, and two more different types of microscopes to show you guys. So we've got scanning tunneling microscopes, which uses a thin metal probe to scan the object, and it has really great resolving power. And then the atomic force microscope actually uses a metal and diamond probe to move along the surface of the specimen and produces a really amazing three-dimensional image. These are on page 61 if you want to look at the pictures. They've got pictures of a protein from E. coli, and then a toxin from Clostridium. So really neat pictures in your book with these newer, very powerful microscopes. So you probably didn't do very much preparing of specimens in A&P. We're going to be doing lots and lots of that in our lab. So for you guys preparing just a typical regular slide, you're going to be using stains in lab. And I put on here, guess what a stain does? Well, it stains things. So you guys have to be very careful in lab with the stains that we use. They will stain your hands, they will stain your clothing, they will stain anything that they come into contact with. So it's very important that you keep the lids on these things when they're not being used, and make sure that you're exercising a special care in operating these stains. So the basic procedure for preparing a specimen is you apply a thin film to a clean slide, and that's called a smear. Then you allow it to dry, then you heat fix it. So when we heat fix things, we're actually going to be using the Bunsen burners, and you're going to run the glass slide over the Bunsen burner 3 times. So, and again, it'd be more helpful if you could see me and I could demonstrate it, which I will certainly do that in lab. But you just hold the slide in your hands. We also will have little clips. I think it's just like clothespins, basically. So you hold on to them with that so you don't burn your finger, and you just run them over the fire. Heat fixing them makes the organism stick to the slide. It also kills the organism, so you don't have to worry about it hurting anybody. Then you're going to apply the appropriate stain. I put appropriate here in italics because the type of stain is going to depend on what we are working with in lab. Then you're going to rinse it off, and then you're going to observe it under the microscope, and your final examination should be done with the oil immersion lens. So you're going to look at it under scanning power, low power, high power. Then you're going to use that drop of oil. You're going to put the drop of oil right on the slide, and then you're going to look at it under the oil immersion lens. You guys will get lots of practice with this in lab. It sounds like a bit of work, and it sort of is, but once you get the right idea and you get the right steps, usually you're good to go. Now, a special type of staining procedure that we're going to do in lab is called Gram stain. It's named after the person, so that's how it got its name, Gram stain. And this is a very important staining procedure in microbiology. So you're going to make your smear, you're going to heat fix it, and then you stain it with a stain called crystal violet, which is a very dark purple stain. Then you're going to rinse it off. You're going to apply iodine. Iodine is called a mordant. A mordant is a chemical that enhances the staining process. Then you're going to wash the slide with alcohol or acetone. Then you rinse it off with water. Then you're going to apply a stain called safranin, which is a red stain. And then you will wash and blot, and then you're going to look at it. So this is much more involved than the typical basic procedure. So you guys will practice the basic procedure first, and then you guys will be practicing the Gram stain. Now, I told you that Gram staining is very important in microbiology, and this is why. Gram-positive bacteria stain purple from the first stain. Gram-negative bacteria stain a reddish pink from the second stain. So all the bacteria stain purple after the first application. The alcohol wash removes the stain from gram-negative bacteria, but not from the gram-positive. So the gram-positive is stained purple, it's going to stay purple. The gram-negative is stained purple, but then it washes away, and then the red stain is applied. So I'm actually going to leave this question open-ended, and it'll also be a good way for me to basically quiz you guys and see if you actually watched these videos. So I'm going to expect everybody to have an answer to this question when we meet back on Wednesday. Why is it so important? I told you it's important. I described it to you. I told you what it's about. But why? Why is this whole gram staining procedure so important? So we will certainly discuss that, and I'll ask for some of your answers when we are back in class on Wednesday. So make sure you have an answer jotted down for that. So another staining procedure is called acid-fast staining. This method is used in identifying a special group of bacteria called Mycobacterium, and they cause two very specific diseases. Mycobacterium tuberculosis causes tuberculosis, or TB, and Mycobacterium leprae causes leprosy, which is probably a disease that you've heard of but don't know as much about as tuberculosis. We will talk about both of these diseases very much in class this semester. So it's a special type of red dye that is taken up by the specific lipids found in the cell wall of Mycobacterium. Mycobacterium are a very unique bacteria, a very unusual group, and they kind of behave a little weird too. And of course they cause two very serious diseases in humans. Some other special stains, some of these, I don't know if we do all of these in lab. I have a feeling we will do a version of all of these in lab, but I'm not 100% sure. So the first thing you can do is you can use special stains to stain for capsules. Capsules don't take up stain because of their molecular makeup, so they actually appear under the microscope as totally blank areas, so totally open spaces. So it's kind of neat to see. Let me see if they have it in the book. Like I said, I believe we'll be doing this in class, but sometimes it's nice to see a picture. Yeah, so this is on page 67, and the capsules are basically just kind of this ring, this blank ring around the bacteria cells. It looks kind of neat. Staining for endospores. This is on the same page. Special types of stains are used to color various types of endospores. We will talk about what endospores are, but they are a structure that bacteria can make, and they make some bacteria very, very dangerous. Endospores is what causes anthrax and tetanus. So endospores are also very important to human health because they can lead to some very serious diseases. And then the last one is staining flagella. Staining the flagella of different bacteria helps to identify which bacteria it is. Some bacteria have one flagellum, others have two flagella, some have 3, 4, some have many, many flagella. So by staining the flagella and actually able to see them and count them, that will help you with the identification of which bacteria species it is. And that's it for Chapter 3. So you have some different types of microscopes that you need to know, and then some different types of stains. This won't be a huge part of Exam 1, which is another reason why I don't mind too much doing the video instead of doing this live in class. I will ask you some questions about the microscopes, I will ask you some questions about the stains, but it won't be a huge part of the test. Definitely Chapters 1 and 4, there will be a lot more material on the test from Chapters 1 and 4. And I will certainly take any questions you guys have about this material, and we're going to talk a lot about this in lab as well, because again, our first lab is on microscopes, using them properly, and all that. So hopefully you guys will all be ready for class and lab on Wednesday. Hopefully you saw my announcement on Blackboard that I did make some pretty drastic changes to our lab schedule in order to fit in with what the other micro teacher does. That way we're on the same schedule. So just, if you already printed out a lab schedule or already looked at it, just get rid of that and go with what's on Blackboard now, because that's the more updated, accurate one. And I will see all of you on Wednesday.